Afternoon all. So, in our previous session till now what did we discuss? It's been 3 days that we have started Cell the Unit of Life. So, yes, we are done with the framework of the chapter cell discoveries, prokaryotic cell, differences between prokaryotic and eukaryotic cell, and then we have started eukaryotic cell, differences between plant cell and animal cell. We have completed with the cell wall. Okay. Let us recall the major aspects of our previous session and then we will move on to the cell membrane, that is fluid mosaic model, which is very important. So, let me check your memory also. In prokaryotic cell, what are the non-membrane bound organelles we have learnt? Ribosomes and inclusion bodies. Can you give me few examples of inclusion bodies? Examples of inclusion bodies. Students from online also you can answer. Phosphate granules, glycogen granules, sulfate granules. Okay, they are surrounded by a protein layer but not a membrane. They are non-membrane bound organelles. And then we have learnt about ribosomes, right? Ribosomes will exist in a specific condition in a prokaryotic cell. What is that condition known as? Very good. Polysome, where a single mRNA will be having multiple ribosomes over it, which is a polysome condition, which is also known as what? Polyribosome. Okay. And in eukaryotes, how can you see it like? Monosome, which will give you what expression? Monocystronic protein expression, whereas in prokaryotes you will have polycystronic protein expression. And then where do you see ribosomes in a prokaryotic cell? We have discussed about 2 locations. What are they? Site of ribosomes in a prokaryotic cell. Site of ribosomes in a prokaryotic cell. So, yes, what are the sites of ribosomes? Ribosomes are attached to plasma membrane and few of them are present in the cytoplasm. Those ribosomes attached to plasma membrane will help in the translocation of proteins, and those ribosomes which are present in the cytoplasm will help in protein synthesis. This is what we have learnt. And 2 more areas where we have to focus from a prokaryotic cell is, what is the role of plasmid DNA? Plasmid DNA. Forgot. What is the role of plasmid DNA? Yeah, helps in conjugation and yeah, it gives a specific phenotypic character to the bacteria and antibiotic resistance. Very important about plasmid DNA. Okay, it gives specific phenotypic character to the bacteria, it gives antibiotic resistance, and it also helps in conjugation. What type of DNA do you see in both genomic DNA and extra chromosomal DNA? What type of DNA you will see, whether linear or circular, in most of the prokaryotic cells? Very good, circular. Most of the prokaryotic cells will have circular DNA, it is double stranded circular DNA. Got it right, ma? And then what are ribosomes composed of? Composed of RNA. Ribosomes are composed of RNA and proteins. And then the major concept from prokaryotic cell, questions will be asked from mesosomes. What are the major functions of mesosomes? Functions of mesosomes. Very good. Increases surface area of plasma membrane. Cell wall formation. Replication of DNA. Formation of daughter cells. Most important function. I am expecting it. Mesosomes are equivalent to which organelle of a eukaryotic cell? Mitochondria. Very good. Mesosomes of a prokaryotic cell are equivalent to mitochondria of a eukaryotic cell. I have learnt it here, right? Because they aid in respiration. How? By secreting out the respiratory enzymes. These are the major points you have to remember from a prokaryotic cell. Non-membrane bound organelles, mesosomes and plasmid DNA. Functions of plasmid DNA. And then, what is the motile structure of a prokaryotic cell? Only motile structure of a prokaryotic cell. Very good. Flagella, it is made up of what protein? Flagellin. What is the microtubular arrangement of it? 9 plus 0 microtubular arrangement. Cilia and flagella, you will be seeing it in a eukaryotic cell. And what are the non-motile structures of a prokaryotic cell? Non-motile structures of a prokaryotic cell. Pili and fimbriae. Okay, pili and fimbriae. You need to revise, keep revising. And then we have learnt on cell wall. Tell me which kingdom system of classification has classified the organisms majorly based on cell wall. We have learnt two kingdom classification proposed by Carolus Linnaeus, wherein all the organisms, be it prokaryotic or eukaryotic, unicellular, multicellular, photosynthetic or non-photosynthetic, they have placed them in plants, if plant kingdom, if they have cell wall. Getting my point or not, everybody? Likewise, you got bacteria into which kingdom under two kingdom system of classification? Plantae. What is the cell wall composition of bacteria? Sangeeta. Peptidoglycan. Peptidoglycan, right? Very good. It has murin. Of what composition? NAG units, N-acetyl glucosamine units it have, right? Next, what is the composition of fungi? Fungal cell wall is made up of chitin. Chitin is also known as fungal cellulose. Okay, and algal cell wall is composed of galactans, mannans, and minerals like calcium carbonate. Plant cell wall is composed of cellulose, hemicellulose, pectins and proteins. Now, when it comes to plant cell wall, we have discussed starting from cytokinesis. How a cell plate is formed? Initially, phragmoplast at the center is synthesized from it is synthesized from Golgi bodies, and then this phragmoplast will move towards the extreme peripheral ends, forming out what? Forming out cell plate. This cell plate will mature into what? Middle lamella. Got it? Likewise, middle lamella is the first formed cell wall layer of a plant cell. Got it right, ma? How it is being synthesized at the center after telophase? After telophase happens, what should happen? Cytokinesis. At the beginning of cytokinesis, at the center what is being synthesized? Phragmoplast. What is it? Phragmoplast, synthesized from Golgi bodies. From the center, it moves towards the peripheral ends, forming out what? Cell plate. Forming out cell plate. Cell plate turns up forming what? Middle lamella. Middle lamella is the first formed layer. It is a common wall layer between two plant cells, okay, and it is rich in what? Calcium pectate and magnesium pectate, okay. That is why in ripened fruits when we eat, right, like guava, sapota and all, we will get that brittle feel because of what? Calcium pectate and magnesium pectate which is present in the middle lamella. Got it right, ma? And then next to the middle lamella, towards the inner side to the middle lamella, what is formed? Primary cell wall is formed. Primary cell wall is thin, extensible, with the growth of cell at the center. What happens? It can break the structural integrity of primary cell wall. Primary cell wall is composed of parenchyma. Next is secondary cell wall. Secondary cell wall is close to the cell membrane. For your idea, let me draw the image. We have discussed it in the previous class, but then again I can see some question marks. So, re-explaining in 5 minutes and then we will move on to cell membrane, okay. As planned, we need to be far ahead, but then I want to go it with your pace. As these are the beginning classes, I want to go it with your pace, okay. Let us assimilate this properly and then we can move on fastly after this chapter, okay. So, the outermost layer is what, ma? Middle lamella. Let us assume at the center this is the cell membrane, okay. Now, inner to the middle lamella, what is formed with a primary cell wall like this, which is single layer, thin, elastic, extensible, made up of what, ma? Parenchyma. Getting Navadip? Yeah. So, this layer is formed inner to the middle lamella, which is primary cell wall. Got it right, ma? Next, inner to the primary cell wall, what is formed? Secondary cell wall layers. Secondary cell wall layers are how many? It can be 2 to 3. Okay, if there are giant plants like gymnosperms, angiosperms, sequoia, eucalyptus, like giant plants, the timber will be formed and the cells will be more matured. So, in that matured cells, secondary cell wall layers can be 3 or more than 3 also. Primary cell wall is single layered, but then secondary cell wall is 2 to 3 layered. 2 to 3 layered. And secondary cell wall is composed of suberin, pectin, lignin, cutin. All these are impervious for water exchange. Okay. Secondary cell wall will have what depositions, ma? Suberin, cutin, lignin. All these are secondary cell wall compositions, right, which will make it very brittle, and then this is impervious for water exchange. Clear, right, till here? So, now if you see, after the formation of middle lamella, next primary cell wall and secondary cell wall layers, from cell membrane, if you see the order, closer to the cell membrane, what is present, ma? Secondary cell wall. Next to the secondary cell wall, what is present? Primary cell wall. Next to primary cell wall, what is present? Middle lamella. Very good. And then middle lamella is the common wall. Middle lamella is present at the outermost layer. This hierarchy you people have to remember. What are the key functions of cell wall? Protection, giving mechanical support and providing proper shape to the cell. Got it? And we say cell wall is dead, but that is wrong. Cell wall is living only, and there is a term when we are discussing in a plant cell, there is a living component in cell wall, they will say. In NCERT also it is there. The only living component of the cell wall is plasmodesmata, they will say. Plasmodesmatal connections. This plasmodesmata will have tubulin like proteins, which are made up of desmotubulin protein. Okay, this is made up of desmotubulin. Plasmodesmata are the connections in between two cell walls, okay, which connect from one pit layer to the other. Direct connections in between two cytoplasms of two different cells, Got it? Understood? This is about cell wall, which we have completed in the yesterday's class, and today let us begin with cell membrane, which is the limiting boundary of the animal cell. Okay? Cell wall is the outermost boundary of a plant cell, which is over cell membrane, but then animal cell limiting boundary is what? Cell membrane. Let us look into the synonyms of cell membrane first. Cell membrane is also known as plasma membrane because it uncovers the complete plasma of the cell. It is also known as semi-permeable membrane because it allows the molecules, solvent molecules directly into the cell. It is known as selectively permeable membrane because it selectively takes what solute particles have to enter into the cell. Got it? Remember these synonyms: semi-permeable, selectively permeable, plasma membrane and cell membrane. Now, there are many models that are proposed to explain the model of plasma membrane, how plasma membrane have to be. There are many models proposed, but no one of them were so realistic. In closer 1950s, after development of electron microscope and all, it is Singer and Nicholson in 1972 have documented the plasma membrane model, known as what? Fluid mosaic model. Fluid mosaic model, proposed by whom? Singer and Nicholson in the year 1972. First, what are the components of a plasma membrane and which plasma membrane they have learnt, they have studied? They have studied the plasma membrane of an erythrocyte or red blood cell. Okay? They have studied the model of erythrocyte or red blood cell. Erythrocyte or red blood cell, right? First, what are the components of plasma membrane? Components of plasma membrane includes, in RBC, what they have documented is, it consists of 52% of proteins, it consists of lipids, proteins and carbohydrates. What are the three components of cell membrane? Lipids, proteins and carbohydrates. Lipids are amphipathic in nature. First we will learn on lipids, right? Lipids here, if you see, they are of what molecules? Amphipathic in nature. What does this mean? It has both hydrophilic head and hydrophobic tail, and lipids are in bilayer. Lipid bilayer we'll call it as, right? It has hydrophilic head and hydrophobic tail. This is hydrophilic head and hydrophobic tail. Got it, right? As it has both hydrophilic head and hydrophobic tail, this is known as what? Amphipathic molecule or amphipathic in nature, right? Hydrophilic head is polar or non-polar, ma? Hydrophilic head is polar or non-polar. This is polar head. Tail is non-polar. Polar head and non-polar tail. How the molecules will be traversing all across the membrane? Look at here. You might not be remembering the terms of hydrophilic, hydrophobic and non, but a simple meaning that you can remember it forever. Phobia means fear, you can know it, right? So this is water fearing group. Okay, this is water fearing. What? Hydrophobic tail. Tails are in water fearing, hydrophobic tails they are, right. So, polar molecules to traverse all across the membrane, they need the help of some carrier proteins and all that we will learn in the transport. Can you see the model clearly? The hydrophobic nonpolar tails are protected from the aqueous environment by hydrophilic polar heads. Got the point? Hydrophobic nonpolar tails are protected from the outer aqueous environment by what? Hydrophilic polar heads. Got it everybody ma? This is about lipids, and how much percent of lipids we can see? 40% of lipids and 52% of proteins, 5 to 8% of carbohydrates. Proteins, how they are classified? We have 3 types of proteins and how they are classified. You can take this down and then we can start with protein. Lipids concept is clear, right? And here the term given in your NCERT that lipids are quasi-fluid in nature. Okay, they are quasi-fluid in nature, which will allow the lateral movement of proteins. Enough. How proteins are classified? Proteins are classified based on the ease of extraction. In biomolecules chapter we will learn, or we have already learned, like how we are getting to know about the biomolecules, how many biomolecules are present, macromolecules, micromolecules, by first what? Taking a tissue, grinding it with mortar and pestle, solventing it, centrifugating it, we will get to know, right. Likewise, to extract proteins based on the ease of extraction, we have 3 different types of proteins. Okay. Proteins are classified on basis of what? On basis of ease of extraction. On basis of ease of extraction, proteins are classified into first peripheral proteins, second transmembrane proteins, and third integral proteins. Okay, peripheral proteins, transmembrane proteins and integral proteins. Peripheral proteins are present at the outer ends of the lipid bilayer, which are easily extracted with the help of detergents. Okay, but then transmembrane proteins are those proteins which can transfer any of the side of the plasma membrane. Okay, it can transpan any of the side or both the sides of the plasma membrane. Okay, transmembrane proteins can transpan any of the side of the lipid bilayer or both the sides. Integral proteins are also addressed as tunnel proteins. Integral proteins are also known as tunnel proteins, as they are embedded inside the lipid bilayer. Okay, they are embedded inside the lipid bilayer, which will transpan both the sides of the membrane. So, as integral proteins are transpanning both the sides of lipid bilayer, can I say all integral proteins are transmembrane proteins? Yes, right. All integral proteins are transmembrane proteins because they are transpanning both the sides of the lipid bilayer. But then all transmembrane proteins are not integral proteins. Important statements to be remembered, so that will not get confused with assertion, reason and statement based questions. This is about the components of what? Lipids and proteins. Now we have rest 5 to 8% of carbohydrates. In what form? Glycoproteins and glycolipids. Carbohydrates are in the form of glycoproteins and glycolipids. Glucose units or sugar molecules adjoined with proteins is known as what? Glycoproteins. This is known as glycosylation of proteins, and glucose molecules or sugar units attaching to lipids is known as glycosidation of lipids. The process happens inside the Golgi bodies. Glycoproteins and glycolipids are synthesized from Golgi bodies and transferred to plasma lemma. Plasma lemma is another name of plasma membrane only. Got it right? I am using all the synonyms so that you will get used to it. This is what ma? Glycosylation of proteins and glycosidation of lipids in Golgi bodies. And this is glycosidation of lipids. Okay, glycoproteins, which is glycosylation of proteins, and this is glycosidation of lipids, that happens inside the Golgi bodies, right? Clear? This is about the composition of plasma membrane. What are glycoproteins? Glycoproteins, glucose units or sugar molecules adjoined with proteins are known as glycoproteins. They are formed in Golgi bodies. Now, amongst the lipids, we have different types of lipids, right? Which type of lipids are abundantly present in the plasma membrane? Okay, phospholipids. And in phospholipids also, phosphoglycerides are highly present in the plasma membrane. It is phospholipids, and in this phosphoglycerides are in highest composition of lipids. These are highest composition of lipids in plasma membrane. Got it right? This is about composition of cell membrane. Shall we quickly recall all this once? Yeah. So, cell membrane, which has many other synonyms. What are they? Plasma membrane, sit straight all. Plasma membrane, semi-permeable membrane, selectively permeable membrane, or plasma lemma. Many models are proposed, of which widely accepted model is fluid mosaic model, because it is more realistic. Proposed in which year? 1972 by the scientists Singer and Nicholson. Let us talk about the composition of fluid mosaic model. It is composed of three main components. What are they beta? Lipids, proteins and carbohydrates. Lipids are amphipathic in nature because they have hydrophilic polar head and hydrophobic non-polar tail. Got it? Hydrophilic polar head is on the outer side. Can you see here? This is the molecules of hydrophilic polar head are outside. These are protecting the non-polar tails from the aqueous environment. Hydrophilic polar heads are protecting what? Hydrophobic non-polar tails from the aqueous environment. What are the highest percentage of phospholipids you can see in the plasma membrane? Phospholipids or phosphoglycerides. In phospholipids, it is phosphoglycerides. Next, proteins. How much composition it is of? 52% of proteins and 40% of lipids. These proteins are of three types. What are they? Peripheral proteins, transmembrane proteins, integral proteins. Peripheral proteins are on the outer side, either sides of the lipid bilayer. They are easily extracted via detergents, whereas transmembrane proteins will transpan any of the sides of the plasma membrane or both the sides. Integral proteins are also known as tunnel proteins because they are embedded inside the plasma membrane. Got it right? So, this is integral protein or tunnel protein, right ma? All integral proteins are transmembrane proteins, but not all transmembrane proteins are integral proteins. Clear, right? And then Carbohydrates, around 5 to 8% they are present in what form? As glycoproteins and glycolipids. Glycoproteins are formed via glycosylation of proteins, glycolipids are formed via glycosidation of lipids. This happens inside the Golgi bodies. Clear till here? This is the composition of plasma membrane. Now let us look into the functions of plasma membrane. Plasma membrane plays major role in cell growth, okay, and it helps major role in formation of intercellular junctions. It plays a key role in formation of intercellular junctions, major role in secretion, endocytosis and cell division. Already one pattern of cell division we have seen, that is plant cell division, how cytokinesis is happening. Animal cell division we will see in the next chapter that we are going to discuss. Amongst this, transport is very important, cell transport, which is facilitated by plasma membrane itself. Cell transport or cellular transport happens in three ways, ma. First is this happens in three modes. First, passive transport. Active transport. Third is bulk transport. Right. So first we will see passive transport. Passive transport is transport of any materials from high concentration to low concentration or high water potential to low water potential. Okay. Transport of any material, it might be gases, liquids. Okay. So transport of that from high concentration to low concentration means it is along the concentration gradient. Yes or no? It is across the concentration gradient. Fine. So passive transport, it is across or along the concentration gradient. It is first via diffusion, means molecules just diffuse, right? This happens mostly with gaseous molecules and neutral solutes. Diffusion mostly happens with gaseous molecules and neutral solutes. Got it right, ma'am? Second is facilitated diffusion. This happens for polar molecules or water molecules. You see here, what is that? Facilitated diffusion. The movement or the transport of polar molecules or water molecules is facilitated via carrier proteins or channel proteins, because it has to travel across non-polar tail, right? A polar molecule has to transfer. It gets adjoined here, but it has to travel all across the non-polar tail. So then this is travelled via what? Channel proteins or carrier proteins. So the transmembrane proteins might be, integral proteins might be, they will act as what? Channel proteins. Okay. So here facilitated diffusion is via what, ma? Carrier proteins or channel proteins. What molecules are transferred? Mostly polar molecules. Example, glucose you can say. And third mode of transport. Is osmosis. Osmosis is also transport of solvent or molecules from high to low concentration or high water potential to low water potential, which is across the concentration gradient. Okay, that is via semi-permeable membrane. One best example of osmosis I can give you is for water molecule, which is traversed across the aquaporins, which is travelled by what? Aquaporins. These are the examples, and 3 modes of passive transport. Does it require any energy, ma? No energy. Okay, and this is from high concentration to low concentration. Next is active transport. The name itself is saying active transport, which is from low concentration to high concentration, means it is against the concentration gradient. This requires ATP. It requires ATP and it is against the concentration gradient. Best example, you can take sodium and potassium pumps. Done. Next is bulk transport. Bulk transport is nothing but any molecules, mostly liquid molecules or solid particles might be, if they are getting transported in bulk, okay, into the cell or out of the cell. It can be influx or efflux. That is known as bulk transport. If cell is consuming lots of lipid particles at a time, that is known as what? Endocytosis. Endocytosis means inside the cell, transport of molecules into the cell. This is of 2 types. One is known as pinocytosis and second is phagocytosis. Pinocytosis and phagocytosis. Pinocytosis also known as what? Cell drinking. Phagocytosis is also known as what? Cell eating. This is for liquid molecules, this is for solid molecules. And then exocytosis. Also addressed as effagi or cell vomiting. Any molecules in bulk, if they are getting exited out of the cell, thrown away from the cell, okay, in group, that is known as what? Effagi or cell vomiting. Okay. So, this is about cell transport, which is very important. Rest all functions, you have the basic idea like cell growth, formation of intercellular junctions, plasma membrane will have its own secretions, endocytosis we have learnt, and cell division. Plant cell division we have learnt. Okay. Clear on cell membrane, plasma membrane, fluid mosaic model. Now, I have to clear out some statements for you which are very important. Okay. Lipids are of fluid in nature. Okay, it will move like this. Lipids are of fluidity in nature. So, that quasi-fluid nature of lipids are allowing what movement of proteins? Lateral movement of proteins. Movement of proteins can be transported because of what? Fluid nature or quasi-fluid nature of the lipids, right. So, you are clear on the composition, you are clear on the functions of plasma membrane. Any doubt anywhere, ma? You can take this down. Students from online, you can have a screenshot. And then we are going to start endomembrane system, if you are clear on this note. Here itself you are writing. Once you are done, just let me know, ma, and then we will learn on the endomembrane system. Done, right? I will save this here, you can write it later as well. So, these are the diagrammatic representations of passive transport and active transport. Just look at it, you will get an idea. Diffusion, can you see? From high concentration gradient to low concentration gradient. Molecules are just diffusing out, which is across or along the concentration gradient. Facilitated diffusion, can you see these are the channel proteins which are facilitating the movement of molecules. Okay. Carrier proteins and channel proteins from high to low concentration again. This does not require any energy, but it requires proteins, channel or carrier proteins. Then active transport from low concentration gradient to high, okay, which is requiring what? ATP. Clear? And then endomembrane system. So, yes, we have learnt that cytoplasm is the main arena of all cellular activities, but then plant cell and animal cell have active compartmentalization of cytoplasm because of membrane bound organelles. Both plant cell and animal cell have single membrane bound organelles, double membrane bound organelles and non-membrane bound organelles. First we will learn on single membrane bound organelles by taking what? Endomembrane system. Endomembrane system, immediately looking at this word, you know, if I have to take 10 PYQs on this chapter and of them 3 to 4 questions asked from endomembrane system, 2 to 3 questions will be on just the names of the organelles. What organelles are involved in endomembrane system? Okay. Why these organelles are placed under one system? Why they are placed under one roof? Why we are learning only these single membrane bound organelles under this heading? Can anybody tell me? Their functions are coordinated. They are dependent on their functions, like one organelle's function is dependent on the other and all of their functions are coordinated manner. This is a teamwork. Okay. So, here if you see endomembrane system single membrane bound organelles include endoplasmic reticulum, Golgi complex or Golgi bodies, lysosomes, vacuoles. Endoplasmic reticulum, Golgi bodies, lysosomes, vacuoles. First endoplasmic reticulum we will see. Okay. Endoplasmic reticulum is rapid revision will be at the end of the chapter, ma. Yeah. If you have to take this as a cell, okay, and this nucleus. Nucleus we will learn under double membrane. 4 components of nucleus I hope you remember. Outermost it has what, ma? Nuclear envelope. Okay. And inside it has nucleolus, nucleoplasm and nuclear pores. These are the 4 components: nucleolus, nucleoplasm, nuclear envelope, nuclear pores. Extensions of nuclear envelope will form a reticulum like structure like this. Extensions of nuclear envelope will form what? Reticulum like structures. Those reticulum like structures are what? Endoplasmic reticulum. Can you see? This endoplasmic reticulum will divide the cytoplasm into 2 compartments. This inside compartment is addressed as what? Luminal compartment. These are the extensions of nuclear envelope from here. Okay. That extensions are creating some space inside, that is known as what? Luminal compartment. And the space away from this is known as what? Extraluminal compartment or extraluminal space. Okay. This is luminal compartment and extraluminal compartment, that is formed by what? Network of endoplasmic reticulum. Endoplasmic reticulum is of 2 types based on the surfaces it have. If the surface is smooth without having any membranes or any organelles over it, that is known as what? Smooth endoplasmic reticulum. But if the surface is rough because of ribosomes, that is known as what? rough endoplasmic reticulum. Okay. And what is the major role of ribosomes? Major role of ribosomes is to synthesize proteins. So, RER will synthesize what? Proteins. If at all, this is RER. Over RER, ribosome is attached like this. Ribosome is attached by what protein? Ribofourin protein. This protein is addressed as ribofourin. And we have seen 70S ribosomes in a prokaryotic cell, and here it has 80S ribosomes adjoined to the endoplasmic reticulum. What does it have? 80S ribosomes. This 80S ribosomes, larger subunit is 60S and smaller subunit is 40S. Larger subunit is adjoined to the endoplasmic reticulum, forming out what? Rough endoplasmic reticulum. Got it right? And then smooth endoplasmic reticulum is involved in the synthesis of lipids. It has many other functions also. Yeah. Once let us look into the functions of rough endoplasmic reticulum and smooth endoplasmic reticulum. You are clear on structure and composition, right ma? You are clear on the structure? Ribosomes, what subunit is attached, through what it is attached, and how smooth endoplasmic reticulums are there. Mostly these are the reticulum structures of lamellar, cisternae like structures, but this will be of tubules. Smooth endoplasmic reticulum will be mostly of what? Tubules. Rough endoplasmic reticulum is purely involved in what? Only protein synthesis, and they will synthesize proteins in the form of vesicles which will be sent to Golgi bodies. Okay. Smooth endoplasmic reticulum primary function is what? Lipid synthesis. In animal cells, this is also involved in synthesis of steroid hormones. Okay, synthesis of steroids. And smooth endoplasmic reticulum will also regulate the concentration of calcium ions. This will also regulate the concentration of calcium ions. Okay, which helps in contraction and relaxation of muscle cells. So, those smooth endoplasmic reticulum structures which are present in the muscle cells is known as sarcoplasmic reticulum. Got it? Smooth endoplasmic reticulum present in the muscle cells is known as what? Sarcoplasmic reticulum. What does it do? It will regulate the calcium ion concentration, which will aid in contraction and relaxation of muscle cells or muscle tissues. Got it right? These are the functions of SER and this is the function of RER. In simple terms, RER will be synthesizing naive proteins, SER will be synthesizing naive lipids, means lipids and proteins are synthesized in the form of vesicles, but they are metabolically inactive. They will get metabolically active in Golgi bodies. How? They will be properly processed, packaged and transported. The events of Golgi bodies we will see in the next class, but I will end the class once we will do few objective and then we will end the class. Yeah, this you can answer now. Assertion reason. Third and fourth question. Try to answer, ma. Third and fourth question. Cells are variable in shape. They may be round, biconcave or amoeboid, columnar or even irregular. Reason. Different cells perform different functions. Assertion and reason are true, but it is explaining or not. No. Okay. So it is what? Second order. Next question. Cell membrane shows quasi-fluid nature. In cell membrane, integral proteins are partially or totally buried in the membrane. Partially? They are totally buried in the membrane. Partially or totally. So what A is true and? Yes. Next question. On shapes of the cell. D. No. Nerve cell is the longest cell. Yes. Option 1. Next. This is also on shapes. So we will move to some other question. Yeah. On cell theory. Navadeep. Good morning. All are correct. Select the right option which relates to Schwann regarding the following statements. Okay. He reported that cells have thin outer layer which is today known as what? Plasma membrane. Cell wall is unique to the plant cell. Yes. The body of plants and animals are composed of cells and products of cells. All the statements are correct. Next. This is also on cell theory. Yeah. Try to answer this question. And one more question on diagram based fluid mosaic model, and then we'll end. First option, we'll see. A is this outer membrane only, which is plasma membrane, as this is animal cell. Cell wall will not be there. So yes, it is plasma membrane. B will be nucleus itself. The labeling is not here, it is here. It is nucleus. C, if you see, it is mitochondria. And D, Golgi bodies. Got it? Next. This question try to answer, ma. Fluid mosaic model of plasma membrane is given. We need to identify it correctly. Second, first. 1 is what? Here it is labeled as 1. Sugar, glycoprotein it is forming. 2 is protein. 2 is protein and 1 is sugar. Next 4 is what? Integral protein. So, this will be wrong, right? 1 is not protein. Sorry, the labeling is wrong, but then we will remember the labelings because we may get the diagram based question, right? Yeah. So, remember carefully, these are the molecules which are the extensions of proteins, right, which is sugar. This is peripheral protein, integral protein embedded inside the membrane, and then phospholipid bilayer, both the sides. This labelings you just remember, okay. So, with this we have completed cell membrane. With this we have completed cell membrane. In the next class we will be starting endomembrane system also. We have done with endoplasmic reticulum, and then we will be starting with Golgi bodies, okay.